Yeah, so ether is a family of compounds which have a carbon attached to another carbon through oxygen. We call this thing in the middle linkage, okay? And over here, it's the ether linkage. It's the ether linkage that is connecting these two parts of carbon together. Now, these are alkyls, can be maybe methyl or ethyl or something else. But they don't always have to be just a hydrocarbon. They can be something else. They can be two strands of DNA. They can be two strands of some other complicated compound, phenol or something. But the linkage is what connects it together. So we usually draw this through a box, that instead of saying that, okay, there is R in there, because R का तो मतलब है कि उसमें सिर्फ carbon और hydrogen है. We say that this is a box. Whatever is in there doesn't really matter, but something is in there. And that is joined together through an ether linkage. Okay, so sugars are all ether linkages. Okay, they all have these ether linkages where oxygen is joining different parts fit together. And we can easily break this linkage by adding simple things like maybe water. Okay, so it's possible to break it. So if you have a sugar which has an ether linkage, it is possible to make ethanol from it, and that is the first reaction that we will be discussing right now. Fermentation. Fermentation is you take any sugar, for example, in this case, I'll take glucose, let's say. So glucose is basically two parts of sugar joined together through ether linkages. So C6H12O6. I take that and I break it down. Okay. And what happens is that I will get ethanol, which is C2H5OH, and carbon dioxide. Obviously, I'll balance this out. And this is 2 times this, I think. Yeah. Yeah.
Sir.
Yes?
Sir, मैं पता कैसे चलेगा हमें ये हमें याद करना पड़ेगा?
ये याद करना पड़ेगा क्योंकि ये एक रिएक्शन है जो कि हमेशा ऐसे ही रहता है. Okay, so you take this thing, we have a sugar and you're breaking it down in presence of a yeast. So you will need yeast, and that yeast will allow us to break this thing down and produce carbon dioxide and ethanol. Okay. What is the role of yeast here?
Sir, it respires anaerobically without the presence of oxygen.
Very good. So this is anaerobic oxygen, which means that you need to make sure there is no oxygen present. क्योंकि अगर oxygen present होगी तो glucose की oxidation हो जाएगी. So -O2 का मतलब है anaerobic conditions. So anaerobic conditions. That's the first thing. Second, you need yeast. Now, why yeast is able to do that is because yeast has a chemical in it which is called zymase. And zymase is a catalyst for this reaction. But here's the thing. You need yeast. मैं एक पैकेट में zymase डाल के ग्लूकोज में डाल दूं और anaerobic conditions में तो ethanol कभी भी नहीं बनेगा, because there is more to yeast than just zymase. There's a whole process in there. So yeast is the catalyst. Zymase is not the catalyst as far as your syllabus is concerned. Okay, so you will write yeast in there. So yeast glucose की anaerobic respiration करता है और उसको तोड़कर energy और glucose ethanol and carbon dioxide produce कर देता है. ठीक है? And then we are able to get that ethanol out, and that is called fermentation. Remember that this will only produce ethanol as the alcohol part. Other alcohols like propanol or methanol or any other alcohol that you want, you will still have to rely on the industrial process. Or industrial process क्या था कि we added steam to alkenes और उससे हमारे पास ये produce हो जाता था. Okay, so that's one reaction where glucose is very useful. Another one is respiration. We talked about this when we talked about sources of energy and how energetics are. So respiration is usually done in presence of oxygen. So you take this same glucose and you add oxygen to it, and it will produce carbon dioxide and water. Okay, so we'll balance this equation. There you go. Once it's balanced, we have this here. So this is respiration. Obviously, there's a lot more to this than just that arrow. Okay, so that's how it goes. And all humans, all plants, all animals do respiration. The opposite of this is photosynthesis. Okay, photosynthesis is an endothermic process because it produces glucose. It starts with carbon dioxide and water, and they produce glucose and oxygen. It's the kind of opposite of this. Like people who study bio know that it isn't entirely opposite. One of them depends on Krebs cycle, the other one has a totally different mechanism. Okay, and we use a catalyst for this. That catalyst is basically chloroplast. Okay, so chlorophyll, I think chlorophyll is the chemical and chloroplast is the part of the cell. Is that okay, people who study biology? Unusually quiet class today. Chale? All right, so we have this thing, we have chlorophyll,
जी सर ऐसे ही होता है.
which acts as a green pigment, which has a green pigment in it. It uses light. There are light systems there, jinko use karke we are able to do this. Water comes from the roots. It's absorbed there. Carbon dioxide, obviously, they absorb it through their stomata. ये सारी detail हम bio में पढ़ते हैं. हमें chemistry में ऐसा उसकी requirement नहीं है. So that's done. Now, the next thing is understanding the nutrients. There are three main macronutrients that we have. So nutrients kya hote hain? Nutrients are basically different parts of our diet that we need to function well. So hamari jo ek healthy diet hai, it has to have 6 macronutrients in there, which are vitamins, minerals, water. ये वो हैं जो कि micronutrients हैं. We need them in small quantities. Okay, but there are some that we need in large quantities, like carbohydrates and fats and protein. ये तीन macronutrients हैं. So carbohydrates, fats, proteins. Carbohydrates technically are polyethers. What does that make you like? What does that mean? Polyether का क्या मतलब है?
Sir, it has a chain of ethers.
Yeah, that's right. It has ether linkages in it, and there is a long chain of these things. Obviously, there are branches as well, but mainly they are ethers that determine this. Okay. Now we have fats. Fats को हम lipids भी कहते हैं. ठीक है? And they have another kind of linkage in them, जिसको हम peptide linkage कहते हैं. सॉरी जिसको हम एस्टर लिंकेज कहते हैं. तो इनको हम पॉलीएस्टर भी कह सकते हैं. Like they are not really called that, but we can say that polyesters. And then we have proteins. Proteins have इनको हम polyamide या polypeptides भी बोल देते हैं. ठीक है, based on what link they have. So in carbohydrates we have the ether linkage, in fats we have the ester linkage, and we proteins we have peptide linkage. तो वो लिंकेज समझना जरूरी है कि वो है क्या. तो ethers में ऑक्सीजन की लिंकेज होती है. There is a part of it, there's another part of it, and they're joined together through this O, which is acting as the linkage in there. It's the ether linkage. Similarly, fats में हमारे पास ester linkage होती है, जिसका मतलब है कि हमारे पास कार्बन के साथ दो ऑक्सीजन इस तरह से ज्वाइन हुए होते हैं, and they join the two parts of the fat together. Whatever those parts are, we'll talk about it in a bit. And in proteins, we have peptide linkage. Peptide linkage is basically this thing. Okay. And there's a box in there, there's a box in there, and these boxes again represent whatever they're made from. लेकिन बीच में जो चीज है ये हमारी पेप्टाइड लिंकेज है. तो ये लिंकेज को समझना जरूरी है, और लिंकेज का स्ट्रक्चर आपको जेहन में होना चाहिए. अगर कहीं पे COO लिखा हुआ है तो वो एस्टर लिंकेज है, कहीं पे CONH लिखा हुआ है तो वो पेप्टाइड लिंकेज है. ठीक है. All right, any questions so far? Now the next thing to understand is what are these boxes? ये boxes क्या हैं और ये किस तरह से इसको connect हो रहे हैं? So carbohydrates are made from small sugars. ठीक है, छोटी छोटी sugar molecules होते हैं जिनको ये join करके हमारे पास carbohydrates बनते हैं. ठीक है, so इस वजह से हम carbohydrates को polysaccharides भी बोल देते हैं. Saccharide मतलब sugar, so these are polysaccharides. And then we have fats. Fats को हम polyesters भी कहते हैं, and they have these.
सर
जी.
सर आपका कार्बोहाइड्रेट्स वाले पार्ट पे बिल्कुल आवाज नहीं आई, आप फिर से बोलेंगे?
Sure. Carbohydrates are basically polysaccharides, which means कि वो sugars को join करके बनते हैं. ठीक है, तो हमारे पास जो भी sugar है उसको हम saccharide बोलते हैं. तो carbohydrates are polysaccharides कि different sugars को join करके हमने इनको बनाया होता है. तो in a way we can say that these are all polymers and they're made from different monomers. Monomers मतलब small structures जिनको join करके ये बनाया जाता है. ठीक है, monomers, there should be N not M. Okay, so what are the monomers for carbohydrates? They're simple sugars. So उन simple sugars में हमारे पास glucose is one of them. Okay, and in fats we have two monomers. There's the fatty acid part and there's the glycerol part. Now, based on these terms here, ol and acid, what can you tell me about these monomers? Fatty acid जो होगा, उसमें उसके compound में क्या चीज जरूर होगी?
सर कार्बोक्सिलिक एसिड.
Yes, very good. And glycerol, इसके OL से आपको क्या पता चल रहा है? What does it have?
एल्कोहल्स.
Alcohol, yes, very good. So fatty acids में COOH जरूर होगा और glycerol में OH जरूर होगा. And then in the protein part, we have acids, okay, which we call amino acid. इसके नाम में दो चीजें हैं जो कि हमें बता सकती हैं what it's made from. The amine part and the acid part. Amine part tells me that it has NH2 in it. Remember amine? And acid part tells me that it has COOH in it. Okay? So COOH and amine, these are the two things that make this up. Okay? So these are the main monomers that make these polymers up. So carbohydrates को बनाना है आपने, तो पहली चीज है कि उनमें ईथर लिंकेज के थ्रू आपने सिंपल शुगर्स को ज्वाइन कर देना. फैट्स को बनाना है पॉलीएस्टर लिंकेज के थ्रू, एस्टर लिंकेज के थ्रू फैटी एसिड्स और ग्लिसरॉल को ज्वाइन कर देना. और प्रोटीन्स को बनाना है तो एमाइड या पेप्टाइड लिंकेज के थ्रू अमाइनो एसिड्स को ज्वाइन कर देना. समझ आ गया? तो ये तो तरीका है न्यूट्रिएंट्स को बनाने का. Okay, so how do you make it? Chemically, the process by which you make them up is called polymerization, and how you break them is called condensation. ठीक है? Sorry, hydrolysis. आप condensation करते हो तो ये बन जाते हैं और hydrolysis करते हो तो ये टूट जाते हैं. Now, does anybody remember what condensation is?
Removal of water.
Removal of water or any other simple thing. Okay, and what is hydrolysis?
Passing electricity.
I'm sorry?
Passing electricity.
No, that's electrolysis. Hydrolysis is when you add water. So condensation is when you remove it, and hydrolysis is when you add it. Okay, so what we are going to do is we're going to add water to break this up, and we're going to remove water to make this. So let's start. Let's start with carbohydrates. Carbohydrates mean, let's suppose I have a simple sugar. Chemically, sugars are basically polyols. उनमें multiple alcohol parts होते हैं. तो मैं simplest sugars होती हैं diols. ठीक है? Diol का मतलब है कि उसमें दो alcohol parts होंगे. So we'll take a simple sugar OH on one side, OH on the other side. Let's say I take another one, another sugar, and just to show that this is a different one, I'll put OH on this side and OH on that side. Now I'm going to join them together. What I'm going to do is I'm going to remove water. So let's suppose on this one I remove OH and on the other one I remove H. और दोनों साइड से आप करोगे तो हमारे पास 2 water molecules बन जाएंगे. और जो भी बाकी part बचा है तो I will just take this one. So this is basically this whole part. Sorry. So I'll take this one, copy it, place it here, and I'll take this one with oxygen, whatever is left over here, copy it, place it over here. And once you join them together, do you see that they have an ether linkage in them? This thing in there, that is ether linkage. And this whole part, whatever you have made right now, that is basically called a repeat unit. And now you will simply copy it. इसके आगे इसके जैसा ही एक और part लगा देंगे आप. And to show that it continues forever and ever, we'll draw lines through these bonds और हमें पता चल जाएगा कि ये बहुत ज़्यादा दफा हो रहा है. So it's continuing, it's happening multiple times, and N means that there are so many of them, both of these.
Sir, ये lines वाला box क्या represent कर रहा है?
Which box?
ये जिस पे lines बनी हुई हैं.
It simply means that it's a different one. Like I can be like, okay, I have one which is this, and one which is this. अब हैं तो दोनों OH के साथ मिले हुए और दोनों diols हैं, लेकिन एक का बॉक्स जो है वो मैंने और तरह का बना दिया और एक का बॉक्स और तरह का बना दिया. Box simply means that there is something in there which we do not concerned with. ठीक है? So just to show that they are different, मैंने उसको different color का कर दिया. So this is how you make carbohydrates. You take simple sugars, you join them through condensation, and because this is produced through condensation, we call these condensation polymers. How do I break it up? How do I go back to making it what it was? तो उसके लिए मुझे ये देखना है कि ये जो मेरे पास प्रोडक्ट बनना है इसमें क्या सिमिलरिटी है. तो आप देखो इसमें बेसिकली दो स्ट्रक्चर्स हैं. एक इसमें ये बॉक्स है और एक इसमें ये पार्ट है. So if I want to convert it back to whatever it was, I will use a catalyst and I will add water to it. Water has OH and H. H will come here and OH will come here. And I have converted it back to monomers. So this is a reversible process. If you do condensation, it goes and becomes a polymer. And if you do hydrolysis, obviously you'll need catalyst for that. And if you do hydrolysis, then they simply go back to being whatever they came from. And this is what happens during digestion. हमारी बॉडी में डिफरेंट कैटलिस्ट होते हैं, वो हाइड्रोलिसिस करते हैं और इसको ब्रेक डाउन करके वापस उसके मोनोमर्स में कर देते हैं. Those monomers are then further broken down to get this energy that we need from food. Okay? Any questions in this? All right, let's do it with acids. So let's suppose I have small sugars. Let's suppose I have this thing. Okay. And I have, let's suppose, glycerol है मेरे पास. ठीक है? Glycerol में usually there are 3 parts of simple sugar in there. So इसमें ऐसे करके box होता है. ठीक है? And these boxes represent that there are 3 parts of sugar in there. और वह ऐसे आपस में join हुए हैं. ठीक है? Let's suppose I have that. and I join it with fatty acids. Fatty acids different तरह के होते हैं. Fatty acids, let's suppose एक fatty acid ये है जिसमें COOH है, एक fatty acid ये है और इसमें भी COOH है, और एक और fatty acid बना लेते हैं, वो ऐसे कर लेते हैं, ये fatty acid है और इसमें भी COOH है. So fatty acids can be of different kinds, and I join them with this glycerol. What happens? I just have to remove water. So मुझे देखते हैं, अच्छा यहाँ से मैं OH remove कर देता हूँ, यहाँ से मैं H remove कर देता हूँ. At this stage when you're in O levels, it doesn't matter whether you remove it from H or whether you remove it from COOH. Okay? आप alcohol side से भी remove कर सकते हैं OH को, दूसरी side से भी कर सकते हैं. As long as you're getting water out, it's perfectly fine. So you will obviously get 3 water out because you have 3 places where condensation is happening. Okay? अब कंडेंसेशन तो हो गया, whatever is left, जो भी आपके पास बाकी बचा है, ठीक है, you just copy it. यहाँ पर उसको copy करके लिखते हैं. So I'll just copy this thing here, and once I join them together, ठीक है, ऐसे यहाँ पर है, I'll join it together. So this joins here, this joins here, this joins here, and I have made a fat molecule. So this is a fat molecule because it has ester linkage in it. You can see that this is ester linkage, COO, 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 and there are so many of them. So ये polyester है, ठीक है? You can also do it with a simple diol. Let's suppose आपको नहीं ऐसा diol चाहिए जिसमें triol है, तो आप ऐसे भी कर सकते हो कि आप different OH इस तरह से रखें और different carboxylic acid रखें. So let's suppose I have dicarboxylic acid and I have diol in there, ठीक है, और इनको मैं join करवाता हूँ. All I have to make sure is that I remove water. So this OH is removed from one side and H is removed from the other one, and I just join everything else together. So this way I'm able to do that. Over here, this box will come because this is the one, and over here, whatever is left over, I'll copy that. So there you go. I copy that as it is. Koi hi-fi, nothing fancy thing going on. You simply join them together, whatever it is. And once you have it, you have basically created a repeat unit. ठीक है? और repeat unit में आपको नजर आ रहा है कि कहाँ पर क्या चीज है. On one side you have this monomer and on the other side you have this whole monomer. और अगर मुझे इसकी hydrolysis करना होगा, if I want to reverse it, I will simply add H and OH. ठीक है? Obviously it will keep on repeating now. तो अगर आपको इसको बनाना है, आप इसको multiple types में बना सकते हैं. आप इसकी दोबारा copy कर लें आगे. ठीक है? And that way you are able to draw another part of it, and this shows that this goes on forever and ever. Okay, so important parts to do here is, first of all, remove water. Okay, then join whatever is left together. Okay, good. Third step, copy that again because you have to show at least 3 repeat units. And fourth step, show lines through the brackets, which shows that it's some polymer, it keeps going forever and ever. Okay, so this thing is called fat because it is made through fatty acid and glycerol. लेकिन ये जो नीचे वाली चीज है, ये तो fatty acid और glycerol से नहीं बनी है, ये तो एक simple diol है और ये simple dicarboxylic acid है, ठीक है? तो turns out we can do it without having to produce fat. हमें हमेशा ये बनाना जरूरी नहीं होता है. हमें अगर हम कोई भी ऐसी चीज लें जिसमें डायोल है और डाइकार्बोक्सिलिक एसिड है, हम उनको ज्वाइन कर सकते हैं थ्रू कंडेंसेशन प्रोसेस, एंड वी विल बी एबल टू प्रोड्यूस polyesters. And polyesters are basically used for so many things. Like they can be used to produce ropes, they can be used to produce बहुत सारे garments, they can be used to produce so many things. Like, और आप देख सकते हैं कि they are basically plastics. So plastics are also made through this. What they have in common with fat is that they are both polyesters. They both have ester linkage in them. One important polyester that you need to know about is terylene, which is industrially made. And all we need for that is we take diol, we take dicarboxylic, and we're able to produce polyester, which is basically monomers joined together through an ester linkage. Okay, now let's try to do this, but with the third one that we had, amino acid. So I have different amino acids. There are 20 different kinds of amino acids in nature. So let's suppose I have an amino. What is an amino acid is it has an amine group on one side and an acid group on the other side. बीच में कुछ भी हो सकता है, like यहाँ पर कुछ भी हो सकता है, आप कार्बन, हाइड्रोजन, ऑक्सीजन का कोई भी स्ट्रक्चर यहाँ पे हो सकता है. ठीक है, तो ये हमारे पास एक अमाइनो एसिड है. On one side you have amino group and on the other side you have an acid group. Okay, so amino acids हैं हमारे पास. So let's suppose I have two different kinds of amino acid. I have one that is like this with lines to show that it's a different one, and I have this. Okay, now I just have to remove water from it. तो मैं यहाँ से water remove करता हूँ. यहाँ से OH हो गया, यहाँ से H हो गया, यहाँ से H हो गया, यहाँ पे OH हो गया. So I'm removing water, and that way I'm able to get two water molecules from it. All right. Now whatever is left, I'll join it together. So on the left side, I have this thing, this whole thing. So I'll just copy it and I'll post it here. And on this side, I have this whole thing here. So I'll copy it and I'll post it here. Sorry. Yeah. And now if I join them together, what I have gotten myself is a repeat unit. This whole thing, this here, this is a repeat unit because this is what's going to combine and make copies of itself. So अब मुझे इसी की copies बनाने हैं और वो आगे चलते जाएंगे. So I'll just make a copy of this whole thing, sorry. Yeah, I'll just make a copy of this whole thing and I'll connect it with itself. तो मेरे पास एक long chain बन गई है और मेरे पास ये protein है. I'll just join it together, draw lines to show that it's going on forever and ever, and to show that there are so many of them. And there you go. We have produced protein. Okay. One important characteristic of protein is कि आप देखो every time you have it, you have every box is joined to nitrogen and carbon, nitrogen, carbon, nitrogen, carbon. That's how it goes. CN, 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 CN. That is the linkage. Okay. And this linkage is called peptide linkage, which is why proteins are also called polypeptides. Now imagine I wanted to do the same thing, but instead of having an amino acid वो होता है जिसमें एक साइड पे amine होता है और एक साइड पे acid होता है. मैं एक चीज ऐसे ले लेता हूँ जिसमें diamine है. Diamine मतलब उसके दोनों sides पर amine group है. Okay. और मैं इसके साथ ही dicarboxylic भी ले लेता हूँ. Let's suppose I take a dicarboxylic. Again, these boxes represent nothing but some organic portion that we do not concern ourselves with at this stage. Let's suppose I have this. I have a diamine and a dicarboxylic. Okay. What happens here? Now this diamine, obviously I'm going to remove water. So H from here, OH from here, H from here, OH from here. I just remove that. And what have I left with? I'm left with this on the left side. And I'm left with this on the right side. So यहाँ पे ये है. And now I'm just going to combine them together. So if I combine them together, I have myself a very neat repeat unit. This thing is a repeat unit because now I can simply make copies of it and I can simply join them together. So this is a repeat unit. Repeat unit वो चीज होता है जिसकी copies बनती रहती हैं. So this I'm going to make a copy here. So let's say I do that. So N with H and the box, then N with H and C with double bond O, the other box, and C double bond O. And this I will keep on showing कि ये चलता जा रहा है ऐसे. Remember कि आपने repeat unit पहली दफा बना के उसकी copies बनानी है. ठीक है. So remember कि repeat unit में अगर N से start होके C पे end हो रहा है तो आपने भी N से start होके C पे end करना है. अच्छा, अब यहाँ पे आप देखो, अगर मैं इसको break down करना चाहूँ तो मैं वापस इसका repeat unit ही पकड़ूंगा और उसको center से break down कर दूंगा. मैं repeat unit देखता हूँ कि repeat unit मेरे पास NH box NH C double bond O box C double bond O बनता है. ये मेरा repeat unit है. मैंने इसको break down करना है, मैं यहाँ से break down करूंगा center से. और होगा क्या कि left side के साथ OH attach कर दूंगा और right side के साथ H attach कर दूंगा और मेरे पास मेरे original monomers वापस आ गए. And that is what happens when you digest these things. Now, obviously, this thing is different from protein. How is it different? In the protein, you can see that every box has CN, फिर box आ जाता है, फिर CN आ जाता है, फिर box आ जाता है, फिर CN आ जाता है. That's the main structure of protein. But with this thing, it's very different. यहाँ पर क्या हो रहा है कि आपके पास box है, फिर NC है, फिर box है, फिर CN है, फिर box है, फिर NC है, फिर box है, फिर CN है. Basically alternate कर रहे हैं. प्रोटीन में CN चलता जा रहा था, यहाँ पर वो फ्लिप करता है हर बॉक्स के बाद, और इस चीज को हम कहते हैं पेप्टाइड लिंकेज. ओके, सो व्हाइल द लिंकेज looks the same, it's slightly different in the way it is overall, like the pattern is different. So this is called peptide linkage, this whole thing. Amide linkage. Peptide is the one that is in protein, and amide is the one that is in artificial structures like this, जो diamine और dicarboxylic से बनते हैं. And these are called polyamides, and polyamides में हमारे पास एक बड़ा important है, which is nylon. Again, nylon के भी बहुत सारे uses हैं, especially in the textile sector. So that is an important one for you to remember. Okay, so these are all condensation polymers, and why they called condensation polymers is because we make them by removing simple things like water from these monomers. Then we join the monomers together, और हमारे पास repeat unit आ जाता है. Then we make copies of that repeat unit to make the whole different this big chain of molecules. ठीक है? So जब भी आपके पास ये क्वेश्चन आएगा कि आपने let's suppose different monomers से एक polymer बनाना है, you will take the monomers, do condensation, remove water. Okay, that's step 1. Then you combine whatever is left together. That's step 2. You have found your repeat unit. Number 3, you copy the repeat unit multiple times, at least 2 times in your exam, to show how this polymer is made. To reverse that, obviously, you'll first identify your repeat unit, then you break it in the middle, and you add H on one side and OH on the other side, and that is how you're able to get your monomers back. Okay? Obviously, the uses of these things, everybody knows about them. I will share notes in which you can read them. And that is what the role of biochemistry is in this chemistry O-levels. Obviously, there are many more applications of chemistry in biology, DNA being the very important one, apart from many others, of course. But we don't study them in O-levels. We will study them in A-levels if you take chemistry or bio, right? Even if you do both. There's another subject in O-levels, combined sciences, which involves all of this. So it's good to know about it. All right.